I'm Oliver Harschnitz. I'm a group leader here at the Neurogenomics Research Center at Human Technopole. ADA-SCID, or adenosine deaminase deficient severe combined immunodeficiency, is a rare genetic disorder. ADA-SCID presents primarily as an immune deficiency and is fatal if untreated. However, in addition to the immune deficiency, ADA-SCID patients also develop neurological deficits. And these neurological deficits include cognitive deficits, learning disability, and hearing loss, among other symptoms. And despite the available treatments, such as enzyme replacement strategies or gene therapy, specifically the neurological symptoms are not cured. And because of this is a still open standing question that needs to be addressed, and also one that we need to find additional therapeutic strategies for. The key objectives of our research proposal are to gain a better understanding in the role of ADA in neurodevelopment in general, and specifically in how ADA deficiency or loss of function in ADA-SCID patients leads to their neurodevelopmental deficits and neurological symptoms over their lifetime. Now, this is really critical to understand better how this works and what role ADA plays, because only then can we design better therapeutic strategies to help alleviate the neurological symptoms that ADA-SCID patients suffer from. And the way we will do this in this project is to use patient cells available to us through a collaboration with Alessandro Aiuti's lab at San Raffaele, and reprogram these patient cells into stem cells. Now, using stem cells, this gives us a great opportunity to then differentiate these specifically in the cell types affected by these patients, so brain cells. And we will do this in a 3D context. So we will differentiate their own stem cells that we then have in the lab into 3D brain organoids, and then look at a single cell resolution, specifically which types of cells in the brain are affected and are essentially diseased by a loss of function of ADA in ADA-SCID patients' own cells. So the strategy we have to approach this project is to use our stem cell technology available to us and then differentiate at scale patient-derived organoids. Now, Elisa Colombo, a postdoctoral researcher in the lab, is really leading and pioneering this project, and she will reprogram from patient cells iPS cells, or induced pluripotent stem cells, which we will then differentiate into brain organoids. And again, this will allow us to understand which cell types in the brain are actually affected. Now, what we will then do is use a really excellent single cell genomics technology to really understand how ADA and the loss of function of ADA leads to a perturbed or diseased cell state. And we can do this then for each different type of cell in these brain organoids, which closely recapitulates the different brain cells of the human brain. One of the biggest challenges of disease modeling, of neurological disease in general, and I think also specifically this is true for ADA-SCID, is the great diversity or heterogeneity that we see in patients' symptoms. Now, this is challenging because if we were to use a single cell line or a single model system, this would not be a fair representation of what all patients really display, the symptoms they suffer from. So the challenge really is to overcome this and to really try and recapitulate the broad diversity of a patient's symptoms in a dish. Now, one way to try and address this is to reprogram first from many different patients stem cell lines, which we will do, and then differentiate from all these patient lines brain organoids. And we hope that, and we expect also to see that different patient organoids will show slightly different phenotypes, as it were. So they will show different disease effects of the mutations that they carry. And then by looking as a whole, looking at the population of all these organoids from these patients, we can better understand the disease as a whole. But we can also then better understand the differences within these patients. I think that's probably going to be the main challenge, the diversity. Our lab is very much interested in leveraging state-of-the-art stem cell technology to study neurological disease and also primary immunodeficiencies that affect the brain. Now, ADA-SCID is in particular a very interesting disorder due to the fact that its primary immunodeficiency is a systemic disease, but the neurological symptoms are actually independent of that. So these neurological symptoms actually occur to the genetic mutation, but not due to the immunological dysfunction. Now, as we see, progress over the past 20 years have really helped cure the immunodeficiency in these patients. And I think this is an extremely important breakthrough, which has been pioneered to a large part by the scientific community also here in Milan and at San Raffaele and at TGET. Now, unfortunately, since then, we've found that these patients, despite having an immunological rescue, still develop neurological symptoms. And this is something we would now like to address, because this is still an important disease burden for these patients. So this is something I think is very important, while we actually see, not only for ADA-SCID, but for other rare diseases, that their initial disease may be cured or alleviated, but then new symptoms over time become apparent. And I think that is clearly the case here for ADA-SCID. Originally, I didn't train as a scientist. I trained as a medical doctor and then specialized in neurology. And while I found really fascinating to understand what these patients would suffer from and try and help patients and treat them, I found it very frustrating that specifically in neurology and for neurological disease, that the therapeutic strategies that we had available to us were often very limited. And we could often actually not cure patients. I think this is true for many neurological disorders. So this really motivated me, and also during my studies and during my PhD eventually, to try and understand better the underlying disease mechanisms that really lead to the symptoms that patients suffer from, also in ADA-SCID, for example, in the hope to be able to better understand the disorder, the disease, and then also in the future to be able to develop better therapeutic strategies, really with the goal to go from bench and the lab to bedside, to the patients and back again, really, to hopefully be able to treat them in the future.